Have you ever pondered over the significance of understanding drug targets and their mechanisms of action? Well, let's set the stage for an intriguing journey into the world of biochemistry. Drug targets, which are usually proteins or nucleic acids in our bodies, play a vital role in the action of drugs. They are the bullseye that drugs aim for to exert their effects. Recognizing these targets and their mechanisms of action is fundamental in therapeutic interventions, leading to a more targeted and effective treatment approach. Now, these drug targets possess unique biochemical features and serve essential physiological roles. For instance, enzymes are a primary type of drug target. They catalyze biochemical reactions within our bodies, and drugs can either inhibit or enhance their activity. Understanding this dynamic interaction between drugs and their targets helps us unlock the doors to advanced healthcare solutions. Stay tuned to dive deeper into the world of drug targets. You're about to embark on an informative and fascinating journey. Let's delve into the fascinating world of oxidoreductases, particularly focusing on monoamine oxidase and cyclooxygenase 1 and 2. Oxidoreductases are enzymes that catalyze the transfer of electrons from one molecule, the reductant, also called the electron donor, to another, the oxidant, also known as the electron acceptor. This process is called oxidation reduction, or redox. Now, let's take a closer look at monoamine oxidase. This enzyme plays a vital role in the breakdown of monoamines, substances that include neurotransmitters like dopamine, norepinephrine, and serotonin. By breaking down these neurotransmitters, monoamine oxidase helps regulate mood and emotional responses. Certain drugs, known as monoamine oxidase inhibitors, can block this enzyme and increase the levels of these neurotransmitters. This can have a therapeutic effect, particularly in the treatment of depression and Parkinson's disease. On the other hand, cyclooxygenases, specifically cyclooxygenase 1 and 2, commonly referred to as COX-1 and COX-2, are key enzymes in the production of prostaglandins, which are compounds that contribute to inflammation and pain. COX-1 is generally present in most cells and tissues and plays a role in maintaining the stomach lining and kidney function. COX-2, meanwhile, is primarily involved in inflammation and pain. Drugs that inhibit these enzymes, known as non-steroidal anti-inflammatory drugs or NSAIDs, can reduce inflammation, pain, and fever. However, it's important to note that while inhibiting COX-2 can alleviate symptoms of inflammation and pain, inhibiting COX-1 can lead to adverse effects such as gastrointestinal bleeding. This is why selective COX-2 inhibitors were developed, to provide the benefits of NSAIDs without the potential adverse effects related to COX-1 inhibition. In essence, oxidoreductases like monoamine oxidase and cyclooxygenase 1 and 2 are not just enzymes. They are vital cogs in the machinery of life, playing significant roles in our bodies, and understanding their function and how they interact with various drugs is crucial in the development of effective and safe treatments. These enzymes play a crucial role in our bodies, and understanding them helps in developing effective drugs. Next, we will explore transferase, tyrosine kinase, and hydrolases, human factor EXA. Diving into the world of enzymes, we find the transferase, tyrosine kinase. This enzyme is like the master switch in cells, regulating cell division, cellular metabolism, cell growth, and cell death. The biochemical feature that sets it apart is its ability to transfer a phosphate group from ATP to a protein in a cell. This process is known as phosphorylation, a crucial step in signaling pathways within cells. When it comes to its physiological role, tyrosine kinase plays a crucial part in many cellular processes. However, when this enzyme is overactive, it can lead to conditions like cancer. Hence, drugs that inhibit tyrosine kinase, known as tyrosine kinase inhibitors, have been developed and are used in cancer treatment. Now let's shift our focus to hydrolases, human factor EXA. This enzyme is a key player in the blood coagulation cascade, a series of reactions designed to stop bleeding. Factor XA catalyzes the conversion of prothrombin to thrombin, a significant step in the clotting process. Its unique biochemical feature is its ability to hydrolyze peptide bonds, which is essential in the clotting mechanism. The physiological role of factor Ka is pivotal in blood clotting. However, when factor XA is overactive, it can lead to harmful blood clots. This is where factor Ca inhibitors come into play. These drugs inhibit the activity of factor Ca, preventing the formation of blood clots that can lead to conditions like deep vein thrombosis and stroke. Understanding these enzymes, their physiological roles, and how drugs interact with them can provide us with a deeper appreciation of how our bodies function and how medicines work. It's like solving a puzzle, where each piece, each enzyme, each drug plays a critical role. And by understanding these pieces, we can better understand the bigger picture, potentially paving the way for the development of new therapeutic strategies. Understanding these enzymes can pave the way for the development of new therapeutic strategies. It's time to discover the world of esterases, AIK, phosphodiesterase 1, phosphodiesterase 5. Each of these enzymes has a unique role in our bodies, and understanding them is key to developing effective drugs. Let's start with H, or acetylcholinesterase. This enzyme plays a vital role in nerve function. It breaks down the neurotransmitter acetylcholine, which is involved in muscle movement, pain responses, and memory. Because of its role in nerve function, AIK is a target for drugs treating conditions like Alzheimer's and myasthenia gravis. These drugs, known as H inhibitors, work by blocking the breakdown of acetylcholine, thereby increasing its availability in the body. Next up is phosphodiesterase 1, or PDE1. This enzyme plays a role in signal transduction, a process that allows cells to respond to different cues. It does this by breaking down cyclic nucleotides, molecules that act as signals within cells. Inhibitors of PDE1 are being explored as potential treatments for neurological and cardiovascular diseases. Lastly, let's talk about phosphodiesterase 5, PDE5 for short. This enzyme is found primarily in the smooth muscle of the corpus cavernosum, a spongy tissue in the penis. PDE5 breaks down cyclic guanosine monophosphate, a molecule that relaxes smooth muscle tissue. PDE5 inhibitors, like the well-known drug sildenafil, work by preventing this breakdown, which can help treat erectile dysfunction. Each of these enzymes, AIK, PDE1, and PDE5, has its own unique substrates and antagonists. Understanding these features is essential for designing drugs that can effectively target these enzymes. For example, AIK inhibitors need to be able to bind to the enzyme's active site, blocking it from breaking down acetylcholine. Similarly, PDE1 and PDE5 inhibitors need to prevent these enzymes from breaking down their respective cyclic nucleotides. Knowing how these enzymes work is crucial for drug design and development. By understanding their physiological roles, substrates, and antagonists, we can better design drugs that will effectively target these enzymes and help treat a range of conditions. Let's now journey into the realm of lysases: DOPA carboxylase, carbonic anhydrase, histidine carboxylase isomerases. Our first stop is DOPA carboxylase, an enzyme that plays a crucial role in the biosynthesis of neurotransmitters like dopamine. By converting L-DOPA to dopamine, it's a key player in maintaining our mood, memory, and motor skills. The drug levodopa, used in the treatment of Parkinson's disease, is a substrate for DOPA carboxylase. It essentially tricks the enzyme into producing more dopamine, helping to alleviate symptoms. Inhibitors of this enzyme, like carbidopa, are also used in combination with levodopa to prevent the breakdown of the drug before it reaches the brain. Next, we encounter carbonic anhydrase, an enzyme that catalyzes the rapid conversion of carbon dioxide to bicarbonate and protons. This reaction is fundamental to many biological processes, including respiration and the transport of carbon dioxide in the blood. Drugs that inhibit carbonic anhydrase, like acetazolamide, are used to treat conditions such as glaucoma, altitude sickness, and certain types of epilepsy. Finally, we have histidine carboxylase isomerases. These enzymes catalyze the conversion of histidine to histamine, a compound involved in local immune responses and regulating physiological function in the gut. Histidine carboxylase isomerases are targeted by drugs used to treat allergic reactions. Antihistamines, as they're called, work by blocking the action of histamine, thus reducing symptoms like itching, sneezing, and runny nose. As we've seen, these enzymes and their substrates and antagonists are at the heart of many treatments for a variety of conditions. By understanding how these enzymes function and how drugs interact with them, we can develop better, more effective treatments. It's a fascinating field where the tiniest molecular interactions can have profound effects on human health. A thorough understanding of these enzymes can lead to the discovery of new drugs. So, let's keep exploring, keep learning, and keep innovating in this wonderful world of biochemistry. We have journeyed through the intricate world of drug targets, understanding their biochemical features, physiological roles, and their substrates and antagonists. From the oxidoreductases like monoamine oxidase and cyclooxygenase, to the critical human factor assay, we've delved into the complexities of how these enzymes interact with drugs to produce therapeutic effects. We've also explored the realm of esterases, namely H-phosphodiesterase 1 and phosphodiesterase 5, and how they play a role in drug action. Lastly, we've touched upon lysases such as DOPA carboxylase, carbonic anhydrase, and histidine carboxylase isomerases, and their unique functions in the biochemical world. Each of these drug targets holds a key to unlocking new treatments and therapies, and by understanding them, we can continue to push the boundaries of what's possible in medicine. But our journey doesn't stop here. Subscribe for more because you won't find me later. Dive deeper into the fascinating world of drug targets and pharmaceuticals with our upcoming videos.